of cell. It controls all metabolic processes and hereditary activities of the cell. The nucleus was first discovered and named by Robert Brown in 1833. The occurrence of a nuclear membrane was first revealed by O. Hartwig in 1893. The nucleus is found in all eukaryotic cells of plants and animals. However, some eukaryotic cells, such as sieve tubes of higher plants and mammalian erythrocytes, have no nucleus. Usually, the cells contain single nucleus mononucleate. However, certain cells may have more than one nuclei. Accordingly, they may be called binucleate or polynucleate cells. The polynucleate cells of animals are called syncytial cells, osteoblast cells. The shape of the nucleus may be spherical, elliptical, or discoidal. In certain cells, the nucleus is irregular in shape. The size of the nucleus may vary from 3 micrometer to 25 micrometer in diameter. The size is directly proportional to that of the cytoplasm. Nuclear size may also be determined by the number of chromosomes or ploidy. The nucleus of the haploid cells are smaller than that of the haploid cells. Nuclear membrane, nucleoplasm, nucleophore, nucleolus. The nucleus is surrounded by a nuclear envelope. This envelope is comprised of two membranes of 510 nanometer thickness. The inner nuclear membrane supports a fibrous sheath called nuclear lamina. The inner nuclear membrane is surrounded by the outer nuclear membrane. The space between the inner and outer membrane is known as perinuclear space. It is a 10 to 50 nanometer wide fluid-filled compartment. The nuclear lamina as a protein meshwork. It has very dynamic structure. The nuclear envelope is perforated by nuclear pores. Each pore has a diameter between 10 nanometer to 100 nanometer. It has been calculated that the nuclear pores account for 5 to 15% of the surface area of the nuclear membrane. There is continuous movement of molecules across nuclear envelope through the pores. The nucleus is filled with a transparent semi-solid matrix known as nucleoplasm or shape. The chromatin threads and the nucleolus remain suspended in the nucleoplasm. The nucleoplasm is composed of proteins, enzymes, and minerals. The nucleoplasm contains several thread-like coiled structures. These are the chromatin fibers. During the cell division, they become thick ribbon-like structure, known as chromosomes. The chromatin is made up of deoxyribose, nucleic acid DNA, and protein. The nucleus contains one or more spherical colloidal structures, nucleoli. The size of nucleolus is related to the synthetic activity of the cell. The number of nucleoli in the cell may be 1, 2, or 4. Chemically, nucleolus contain DNA of nucleolar origin. 4 types of RNA, 70 types of ribosomal proteins, RNA binding proteins, and RNA splicing nucleoproteins. Ribosomal subunits are synthesized in the nucleolus. Initiation, production, and maturation stages of ribosomal formation happen in three distinct regions of nucleolus. Chromosomes. The chromatin fibers get condensed into chromosomes. During cell division, they are capable of self-reproduction and they play an important role in heredity. The nucleus was first observed and described by Carl Nagy in 1842, and the nuclei of plant cells, chromosomes, and their role in cell division was first explained by Later, in 1873 and 1885, Wendell and Bowery reported that the number of chromosomes for each species is constant. T.H. Morgan and H. Muller in 1922 revealed the occurrence of nearly 2,000 genetic factors on four chromosomes of Drosophila. And in 1924, Robert Fulgen showed that chromosomes contain DNA. The number of chromosomes is constant for particular species. The reproductive cells, such as sperm or ovum, have set of chromosomes, and it is known as the haploid. It is also known as genome. The somatic or body cells contain two haploid sets or genomes and are known as the diploid cells 2N. The diploid condition is arrived at by the union of the haploid male and female gametes and sexual reproduction. Number of chromosomes. Paramecium 30 to 40, Hydra 32, Housefly Musca domestica 12, fruit fly Drosophila species at pigeon, Columba Livia 80, Gorilla 48, men Homo sapiens 46. The size of chromosome can be measured during metabolic metaphase. It may range from 0.25 micrometer to 30 micrometer. Types of chromosomes: telocentric, acrocentric, submetacentric, metacentric. The shape of the chromosome changes from phase to phase. Each chromosome has clear zone known as centromer or kinetochore. Along their length, the centromer divides the chromosome into two parts. Each part is called the chromosome arm. Thus, according to the position of the centromer and nature of the chromosome arm, the chromosomes may be telocentric, acrocentric, submetacentric, and metacentric.